Alrighty, hi guys, welcome to 3.2, the cell cycle. Now this is a fairly long video, so I would buckle up. Alright, let's do it. Okay, so unlike prokaryotes, eukaryotes are far more larger and more complex. And so the eukaryotic cell cycle contains three distinct phases. Interphase, which occurs between cell division, and within interphase, this stretch over here, is the gap 1 or the G1 phase, the synthesis or the S phase where DNA replication occurs, and the gap 2 or G2 phase. Okay, after interphase is the mitosis phase where the nucleus divides, and that contains prophase, metaphase, anaphase, and telophase, forming the acronym PMAT. After mitosis, we have cytokinesis, which is the division of the rest of the cell. Now, cytokinesis and mitosis together forms the cell division part, and then the rest is interphase. Okay, let's go into each one with a little more detail. Let's start with interphase. So interphase, the word inter means between, and then the word phase is phase. And so interphase is the phase in which the cell actually carries out its regular function, its normal duties as whatever that type of cell is, whether it's a heart, a lung, or a brain cell, or whatever. It grows and it's going to develop, and it also replicates its DNA. In the mammalian cell cycle, which is about 24 hours, 22 of those hours are typically spent in interphase, which again varies between different types of cells. So the three stages of interphase starts with the gap 1 or the G1 phase. Okay, so this occurs straight after the previous cell division. And so the cell, after it has just divided into small halves, is now going to grow and double in size to become back to its original size by producing more organelles, more proteins, and then expanding the plasma membrane. During this time, it's also going to perform its vital functions, and that is typically the longest part of interphase, right? Now, a bit of a sidestep. If the conditions are not right to progress into the next stage, the cell actually might stop its cycle and enter into what we call a resting phase or a G0 phase, okay? And you can see there, this kind of arrow splits off from G1 before it goes into the S phase. It might split off into the G0 resting phase. That resting phase is where the cell is going to continue to function normally without any changes to its internal structure, and it's not going to progress into its synthesis phase, right? If the resting phase is reversible, it's called quiescence, but if it's permanent, in other words, the cell is never going to divide, it is senescence, okay? So for example, right, your white blood cells, the cells that are responsible for your immune response, they are going to enter into quiescence when there is no pathogen available, and then when there is an invading body or a pathogen, it's going to revert back into its regular cell cycle stage and then attack the pathogen, right? So that's what this animation looks like. That's the, like a worm or something like that that's invading the body, and then all of a sudden your white blood cells are going to start attacking it and pump out more and more white blood cells to kill it. Your red blood cells, on the other hand, as they mature, they actually develop in the bone marrow, and when they reach the erythroblast stage, they begin to lose their nucleus and other organelles and turn into a red blood cell, an erythrocyte, and that losing of organelles means that the red blood cell will begin to enter into senescence because it's never going to divide. A mature red blood cell does not have a nucleus, and it only needs to transport oxygen in its hemoglobin molecule at the very center. So that means that the red blood cell is going to stay in the resting phase and stay in senescence, okay? Now, the rest of interphase consists of the S or synthesis phase and the gap 2 phase. In the synthesis phase, the chromosomes that are present are going to replicate and duplicate in number, and the DNA replication process occurs in this particular stage. In other words, the DNA that the cell has is going to double in number in the chromosomes, and the replicated chromosomes, which are now called chromatids, which are joined together at the centromere in the middle, are going to double in the amount, right? At the end, the total number of chromosomes, what we call the ploidy number, are still the same, but the amount of DNA in each chromosome is going to double, right? So here is an example of DNA replication, right? This is a chromosome on its own, and then when it goes through the DNA replication phase, you're going to get double the amount of genetic information, but it's joined together in the middle here at the centromere. And so technically, you still have 4 chromosomes, 1, 2, 3, 4, but each one has almost an identical photocopy that's joined together, right? And so you have 1 chromosome, 1 chromatid at the start, but then by the end, you have 1 chromosome with 2 genetically identical chromatids, okay? The gap2 phase, the G2 phase, occurs right before it's going to divide its nucleus, and during this stage, the cell is going to undergo more growth and prepares all those necessary materials so that mitosis and cell division can occur, okay? Let's take a look at the DNA replication stage in more detail, and I'm going to forewarn you that this is a fairly complicated process, and I want you to kind of tune in and just make sure you follow it along carefully. And if at any stage you're not sure, just kind of go back and just watch sections of this video again. So the DNA molecule has a double helix structure, okay? That means it has kind of like a twisted ladder rung with an outside and inside, and you can have the outside kind of forming this kind of double staircase kind of thing, and then the inside are your nucleotides, which is your A, T, G, and C, which are going to join together like rungs in the ladder in between the two outside, okay? Now, that particular structure of DNA means that if you have 1/2 of the strand, that 1/2 can become a template for a new DNA molecule to form. And so DNA having this unique structure means that you can synthesize new strands of DNA based on that particular half. What you have to do is you just have to unravel it and then replace the sections in the middle and reuse those halves as templates for new ones. Now, how does that work? Well, the parent DNA strand is going to be unzipped, much like a zip in a jacket, by a particular enzyme called helicase at the replication fork. So in the diagram, this is the DNA helicase, and then it's going to unzip and unravel that particular section of DNA. And then from there, this top half here, that is going to be opposite to this bottom half, and a RNA primase molecule is going to come along and it's going to attach the first couple of nucleotides to that, right? After the RNA primase attaches the first molecules, the first nucleotides to it, another molecule called DNA polymerase, this brown one here up the top, and then this brown one here down the bottom as well, those DNA polymerase molecules are going to attach themselves, and they are going to assemble the opposite half of those DNA strands, okay? So you've unzipped them, and then they're going to run across this particular zip section, and it's going to start attaching new nucleotides to the other half of the DNA, and it follows a particular type of pattern, right? Adenine, the nucleotide represented by the green molecule here, that's always going to pair with thymine, right? In other words, every time a green appears, you're going to see thymine appear on the other half of the yellow. So this one's green and this one's going to be yellow. This one's yellow and therefore it's going to be green because they are always complementing one another. Cytosine, which is C, in this kind of bluish kind of color here, that's always going to pair up with guanine, which is red. And so you can see, once again, there's guanine, and opposite guanine is going to be cytosine. There's cytosine, and opposite cytosine is going to be guanine, right? So DNA polymerase is going to assemble that in that particular order. Every time it reads a C on the top half, it's going to attach a G. In the bottom half, every time it reads an A, it's going to attach a T. And every time it reads a T, it's going to attach an A, and so forth, right? It's going to keep doing this until, by the end of that particular process, it's going to have a new double helix strand up the top and a new double helix strand down the bottom, giving you two identical DNA molecules based off the original DNA molecule strand, right? Now, there are complications to this process. You're going to immediately notice in this diagram that the top half looks very fancy, but then the bottom half looks a little bit kind of conjuncted and kind of randomish, right? There is actually a reason for that, okay? Complications occur during DNA replication that makes the process a bit more cumbersome depending on which top half or the bottom half that you're in. The DNA molecules, they are not running exactly parallel to one another in the same direction. If you were to think of DNA as being two molecules, right, they actually run in opposite directions from one another. In other words, chemically speaking, the molecules are actually running in opposite directions, right? If you were to look at this particular figure here, right, it turns out the phosphate, which is at the top, which joins to the sugar molecule, S for that sugar molecule, right, in the middle, and then that in between ladder rung there, those are the nucleotides. Well, the phosphate and the sugar molecules actually face a certain direction. And that particular direction is what we call the 3' to 5' end, right? An easy way to identify it is that the arrow from the sugar molecule always points to the 5' end, right? The arrow points to the 5' end. So one strand of the molecule is actually assembled going towards this direction, but the other strand in the original template is actually pointing in the opposite direction, right? Now, in its normal life, that doesn't present too much of a complication, but when it comes to replication, it does actually have a particular effect. Because that DNA polymerase molecule, that miracle molecule that starts to assemble the second half, the new DNA strand, it only moves from 3' to 5'. That means on this particular top template here, the 3' to 5', right, that is what we call the leading strand. This is really easy for the polymerase molecule. The reason for that is because the molecule, as the DNA helicase unzips, so imagine the zip going this way, and then, this little particular section, that's going to unzip next. As the molecule moves this way, on the leading strand, the top strand from 3' to 5', the DNA polymerase simply follows behind the helicase, right? Because it moves in that direction. So it follows it along, and as it follows it along, it's going to attach those new nucleotides along the strand, and it's simple. It's very easy. It just moves along this kind of conveyor belt, follows the DNA helicase, which is the unzipping molecule, and it's going to attach the new Lego blocks of nucleotides along the way. The problem actually happens in the lagging strand, right? Because DNA polymerase moves from the 3', which is over here on the bottom, over to the 5', it's actually moving away from the source of the zip, right? So here's a problem, right? The DNA helicase unzips here, but the polymerase actually moves this direction. So it does this kind of awkward thing where it only attaches a few at a time, and then it actually has to kind of pop off, comes back over to the other end, and then reattaches a few more along this end, and then it pops off, and then it comes back along here and attaches a few more along this end. And all the while, that primase molecule has to actually kind of, reprime the first couple of molecules so the DNA polymerase can take over and attach portions at a time. So on the leading strand, it's easy. It's just one long molecule that's really good. And then on the lagging strand, it's really complicated because you can only do sections at a time here, sections at a time here, sections at a time here. And then another molecule has to come along and it has to kind of basically restitch those in-between molecules that the primer has attached, and then attach and turn it all into a new strand. So a really kind of, really complicated and annoying process in the lagging strand, but a very straightforward process in the leading strand. So all of that, the purpose of all of that is to now give you two DNA molecules that are exactly identical, so that when the cell later on undergoes division, it's going to have enough DNA so that new cells are going to have the same amount as their original parent cell, okay? And that'll become useful later on when I talk about mitosis. Now, before we move into mitosis, I do need to kind of give you guys an idea of chromosome structure. We have different terminologies for how the chromosome is structured, and it's useful when you're looking at mitosis and cytokinesis to understand what the names actually mean. So the DNA at this stage is packed very tightly inside of a cell in order to both protect the DNA and make it easily accessible, right? It's like a library, right? You need to have books, and the books have chapters, and chapters have pages, and so forth, right? This allows approximately 2 meters of DNA to be compacted into only a few micrometers, which is very remarkable in my opinion. The DNA is packed together with these proteins called histones, right? So the DNA, they pack into these nucleosomes, and then they pack around these kind of protein molecules called histones. And when you pack them together, they're really tightly coiled like rope into what we call a chromatin fiber. This chromatin fiber normally is actually too thin for you to see in a microscope. So they are invisible essentially, right? However, during that DNA synthesis phase, the S phase, the chromatin strands are going to be replicated, and they're going to form chromatids, right? And they become a little bit more tightly coiled during mitosis, which makes them more visible, right? And so here is one chromosome strand composed of one chromatid, right? When it goes through the DNA synthesis phase, you're going to get one chromosome strand composed of 2 exactly identical chromatids, what we call sister chromatids, right? And then what happens is when the chromosomes condense, they form this little X pattern, right? And so the 2 identical chromatids are going to coil themselves around again, join at a particular center called the centromere, and that forms one chromosome, right? So notice how it's still called a chromosome, but it has a different shape. Originally, when it is in the S phase, the chromosome is one long straight strand. However, when it goes through mitosis, it is in the shape of an X. So you have one chromosome with 2 sister chromatids, but they are now joined together at the centromere. And then after mitosis occurs, you will actually have 2 chromosomes separated from one another from the middle, and then each one has one chromatid. So you can see there, chromatin is that long invisible fiber, chromatid is a tightly coiled visible fiber, and then a chromosome is the name given to both the long strand of double chromatids and also the X-shaped during mitosis, which is what we will look at next. Okay, let's look at mitosis, right? If you want to take a break, you might want to pause this video, maybe go grab a drink, and then when you come back, take a very deep breath. Here we go. Mitosis is when the nucleus divides, right? It is the name specifically given to when the nucleus within the cell is going to divide into 2, right? There are 4 stages, right? And in this little animation, you can see how it happens. And from the surface, it looks remarkably like binary fission, but there are intricate happenings that occur in each phase that you guys need to be aware of, okay? So there are 4 phases. Prophase, and the word pro means before. Metaphase, the word meta means adjacent. Anaphase, the word ana actually means up. Don't know why. And then telophase, telo meaning last or end, right? So it forms the acronym PMAT to help you guys remember. And roughly this is what it looks like. This is a cell under a microscope undergoing prophase. This is it undergoing metaphase. This is the cell undergoing anaphase. And then this is the cell undergoing telophase. And then you can see there, and that's how it transitions between each section. Now, let's start with prophase. In prophase, the nucleus, the chromatin fibers in the nucleus, it begin to shorten, and they're going to condense into chromatids. therefore becoming visible. Chromatins were invisible, chromatids are now visible. The chromatids are going to condense again to form your chromosomes, which forms that X shape that I was talking about in the previous slide. Now, outside of the nucleus, right, in the cytosol, these organelles called the centrioles are going to start to migrate over to the edges of the cell, and they're going to start releasing microtubule fibers that are going to radiate and form a giant scaffolding network called a spindle, right? That network, right, looks a little bit like this. So there's your centriole, and the centriole kind of looks like two very thick spaghetti straps, just kind of like crisscross with one another, forming a centrosome. So a centrosome is made up of two centrioles, and then they sit on opposite sides of the cell, and they release this kind of scaffolding, like a spider web, that's going to extend outwards towards the middle, right? The nucleus, that nuclear membrane, that's actually going to begin to dissolve, and you're going to start losing the membrane that holds all of the DNA in prophase, right? Here is a micrograph of a cell in interphase, and then here's a cell starting to undergo prophase. You can see that the strands are now becoming a bit more visible. That stain is now becoming more visible in individual strands because they can be seen, and then they're going to condense, and then you're going to start to see this nucleus disappear slowly, right? Now, the next phase is what we call prometaphase. Now, prometaphase occurs between prophase and metaphase, which is the next one, but technically it's part of prophase, right? By now, the nuclear envelope, that shell of the nucleus, is fully dissolved, and the chromosome is now exposed to the rest of the cell. So here's the cell, and there is no more nucleus, and it is only got chromosomes sitting kind of around the center of the cell, right? During this phase, the prometaphase, the kinetochore, which is a protein filament, begins to develop around the centromere of the chromosomes, and allowing the centromeres to attach to the spindle fibers. That means that the scaffolding attaches themselves to the chromosomes, right? And then by now, the cell is ready to go into metaphase. In metaphase, you have a fully dissolved nucleus, and you have your chromosomes start to line up at the equator of the cell, in the very center of the cell, what we call the metaphase plate. And they attach to the spindle fibers, right? It kind of looks like this, right? All the chromosomes, the X's, are lined up kind of horizontally at the X's, and then the chromosome, the centrioles have these fibers that are going to attach to the very center of the chromosomes, which is the centromere, right? In a micrograph, you can tell when a cell is in metaphase because all of its chromosomes are in the middle, and then there's your spindle fiber, and you can't see it, but that's where the centrosome would be, and they're all attached to the chromosomes. Now, here's the important part in understanding metaphase, right? That chromosome X, it's going to lie horizontally because the top half, that top half is going to be genetically identical to the bottom half, right? And so you want to lie it horizontally because that is a identical strand to that one, and later on you're going to want to split them away from one another, right? So that each chromatid, which is identical, is going to face away from each other like so, and that's going to separate. In anaphase, once it is fully lined up, the spindle fibers are going to contract. That scaffold is going to start to pull the centromere apart, and it's going to split up the sister chromatids, right? So there's the animation, right? So that's why you want it to lie horizontally along the equator, because those identical parts are going to be pulled to opposite ends of the cell, which means by the time it splits, each half of the cell is going to have identical chromosomes. Here is a micrograph of anaphase occurring, and you can see the middle, there's now a gap, and you can see the centromeres are now starting to shorten, pulling the chromatids away from one another, right? And you would assume that the DNA in this half of the cell is going to be identical to the DNA in this half of the cell. The last phase is telophase. Once the chromosome arrive at the poles, they are going to start to reform their tight clusters, right? the spindle fiber is going to begin to disassemble because it's no longer necessary, and you're going to get a new nucleus forming around each half of the cell, creating two nuclei within the same cell. The chromatids are going to then uncoil and turn back into their invisible chromatins, and then, kind of during telophase, cytokinesis is going to start to occur, which is the next phase that we're going to talk about, right? So a cell going through telophase starts to look like this, right? It's fully pulled them across to the cell, a new nucleus is going to occur, and then you're going to start to see this little pinching action occur right around the middle. And so you can see it's one very elongated cell that has a slight pinched center, but tight clusters of invisible chromatids, sorry. And then the centrioles losing that spindle network that was originally there. And cytokinesis typically happens around about the same time, and you're going to start to see the rest of the cell pinch, right? Cytokinesis, you can kind of derive the meaning from the name. Cyto referring to cell, kinesis means to move, right? And so cytokinesis is the division of the rest of the cell, the cytoplasm, to form your two daughter cells. So now the mitosis process has finished. You've got two identical nuclei within the one cell, and the organelles are going to start to be kind of divided between the two cells. It looks a little bit different in animals from plants and fungi, and in animals, this is kind of what it looks like, right? So when you get these tight clusters and telophase is finishing, and cytokinesis begins to occur, you begin to see the plasma membrane pinch right in the middle, and that pinching of the plasma membrane is called cleavage. When something cleaves, it's cut, right? And so that cleavage furrow that occurs in the middle is two plasma membrane walls being kind of formed, and then eventually the cell's going to split into two, right? During that time, all the organelles are going to kind of divvy themselves up between the two sides, and then eventually they're going to pinch in half and form your two daughter cells, much like binary fission. In plants and fungi, however, you've got a slightly added problem. The primary reason why it's a little bit different is because plants and fungi have a cell wall. Now remember, the cell wall is an additional organelle that reinforces the cell, giving it structure and rigidity. But in order to form this cell wall, the cytokinesis process looks a little bit different, right? So a new plasma membrane and a new cell wall needs to be formed. That primitive cell wall is called a cell plate, where you have these little vesicles gathering, and then the cell plate starts to form in between the two cells, and then the plasma membrane forms around those as well. And then as it grows, it solidifies. It's kind of like cementing between the two cells, and then eventually you get your two smaller cells occurring in between your original cell plate, right? And that's where your new cell wall forms. Okay. Okay. So here is a summarized version of that particular process of mitosis, and you can see how it progresses from prophase, prometaphase, metaphase, where it's in the middle, and then anaphase along the pulling of the poles, and then telophase with that new nucleus forming, right? Here's an animation showing what's kind of happening in this particular cell, and I just want you to pay attention to how it begins to split off, and then you can see there's your phases, and anaphase and telophase, giving you two distinct cells, okay? Yeah, that's it for this particular video. I hope you guys enjoyed that, and I will catch you guys on the next one.